All right. Welcome, Mr. Peltz. First off, could you just tell us a little bit about what you do here at Stanford and on like a day-to-day basis type
Okay.
thing?
I'm a professor here in the medical school, and my area of interest is in the use of genetics and genomic technologies to discover new therapies for disease.
Oh, great. So we've been learning a little bit about kind of gene sequencing and stuff related to that. And I was hoping you could tell us a little bit about your computational genetics and how that is able to like analyze the genome sequence in like a more efficient and fast way than just traditional methods.
Sure. What we do is we use inbred strains of mice to identify the genetic basis for differences in responses, either physiologic responses or disease susceptibility. There's over 400 different inbred mouse strains, and they differ in a variety of responses. You can look at a number of diseases. And what we do, for example, is we can addict mice to narcotics. So we can give mice doses of morphine. After 4 days, they become addicted to the morphine. And what we do is we looked at about 20 different strains, found that some strains became addicted and other strains didn't. So the question is why? What makes one strain resistant and another susceptible? So we've done that for other diseases, but I'll use the narcotics just as a clear example. So what we did is we've sequenced the genomes now of 23 inbred strains. Each genome is about 3 gigabases, 3 billion base pairs. We generate terabytes of sequence and then analyze it. It's called next generation sequencing. So for these 23 strains, we built a complete genetic map of how one strain differs from another and what the base pairs are that are different. And then on top of it, we invented a computational method where we organized the patterns of genetic variation into these things called haplotype blocks, which are just ordered structures for genetic variation. And we developed a novel computational method that you can input the phenotypic data, the differences in the responses, and it maps it onto genotypic differences and says, look over here, this is the likely cause of the difference. So to go back to our narcotic example, we identified a receptor where the mice had different genetic sequences. We took a drug called ondansetron, commonly used drug to prevent nausea and vomiting, but it had never been used for drug addiction. And we showed in the mice, if we pre-treated them with ondansetron, we could block them from becoming addicted to the morphine. So you might say, okay, who cares? It's just a mouse thing. But what we did was we brought in Stanford students, Stanford undergraduates. They were our normal controls. Now, you know Stanford undergrads are not normal, but we'll call them normal for our purposes.
Right.
We gave them morphine and showed that by pre-treating them with ondansetron, we could block them from going into withdrawal. So now we're about to start a $5 million study at multiple medical centers here in California to see if we can prevent narcotic drug withdrawal in babies born to narcotic-using mothers. About half to 3/4 of those babies, which amount to about 2 to 3% of births in California, they go into withdrawal. They end up in intensive care units for more than a month. So if we can prevent withdrawal, that's a pretty major accomplishment. So it's an example of how little silly studies in mice can actually lead to new therapies.
Right. Yeah, that's really interesting. Kind of something along those lines. Recently, we've been studying and learning about drug therapy and kind of the process of a drug going from like testing, maybe testing with mice to human trials to the long process of getting a drug FDA approved. Can you talk a little bit about what it's been like to transition from mice to humans and what kind of complexities might have arisen or difficulties or frustrations?
Sure. It's actually interesting. Mice, the way I describe it is pretty much everything that you see in a person in terms of basic physiology, you can mimic in a mouse. In terms of the narcotic drug study, we're entering what would be the equivalent of a phase 2B.
Phase
Do you
2.
remember the different stages?
Yeah, we just started talking about this.
Okay, we're phase 2B in the sense that we're going to be looking for efficacy responses, seeing if we can prevent drug withdrawal in the babies, and we're going to be doing it at a multi-center level. So it's probably a phase 2B.
Okay.
If it works, we don't really need a phase 3 simply because the safety database, a big part of what phase 3 is establishing a safety database. You want to test it in about 1,000 to 10,000 people.
Yeah.
And that's already been done. So pretty much if this works, and it'll take about 3 to 4 years to test it, then we'll know, and then it can be used to prevent drug withdrawal in babies, and that's a pretty cool thing.
Yeah.
It's called the narcotic abstinence syndrome.